Hi, good evening, good afternoon, good morning, everybody. It is 6 p.m. in Paris, 6 p.m. in Heidelberg, 6 p.m. in Salamanca, 5 p.m. in London, noon in New York. And it is my great pleasure and honor to welcome you all to this special live webinar entitled Novel Agents in Myeloma, Different Targets and Mechanism of Action. I'm Mohamad Mohty from the Sorbonne University and Saint-Antoine Hospital in Paris, in France, and these are my disclosures. For this educational activity, I will be joined today by two superb faculty, namely Professor Mateos from the University of Salamanca in Spain, and Professor Marc Raab from the Heidelberg University Hospital and the German Cancer Research Center in Heidelberg in Germany. This is a very high-level educational activity, so we do have some ambitious learning objectives. So after participating in this webinar, you will be able, hopefully, to describe the most frequent antigens currently used in myeloma therapy, both for the design of monoclonal antibodies and cellular therapies, to understand the rationale behind targeting such antigens and the mechanism of action of the targeted agents, and last but not least, to highlight the potential advantages and disadvantages of each target and summarize some key results. So we have really an excellent agenda where Professor Mateos has kindly accepted to give you an overview about targeting Bcma, which is really a very popular target, whether for CAR T cells or bispecific antibodies. Then, of course, we will have some discussion, and it will be followed by Professor Raab, who will discuss CD38 and SLAMF7 as targets. And these are also extremely important targets because I would kindly remind you that this is where we have already actually antibodies, monoclonal antibodies, which are approved in the field of myeloma. And of course, after the different talks, we'll have some discussion. This is a highly interactive educational activity. So we would love to hear from you. You can interact throughout this webinar by answering the polling questions, and I'll come back to this in a couple of seconds. You can send your questions, your comments, your suggestions at any time. And if you want to participate in the poll, you have to exit, of course, the full screen mode, and the polling will be on the right-hand side of the screen next to the video. Of course, please do not forget to complete the evaluation form at the end of the webinar, but I will remind you this later on. I'd like also to acknowledge that this educational activity is made possible thanks to an educational grant from GlaxoSmithKline. In order to measure the learning and to assess the objectives of the educational activity, as I mentioned, we'll share with you a few questions, but the correct answers will be revealed during the post-assessment. And this is the first question, so please get ready to answer. Question number one, and I will read it. For CAR T cell therapies, which of the following statement is incorrect? So you have to find the wrong statement. Answer number A, they are less effective than bispecific antibodies or antibody-drug conjugates for the treatment of multiple myeloma. Statement number B, treatment only requires a single infusion. Statement number C, not all patients with refractory multiple myeloma are eligible for treatment. So please vote. Thank you for voting. Question number 2. CAR T cell therapies for multiple myeloma are currently under development for which of the following target antigen? Is it PD-L1? Remember, checkpoint inhibitors are really important. Is it SLAM-F7 or is it CD40? Please vote A, B, or C. Next question, number 3. Trispecific antibodies, not bispecific, trispecific antibodies have 3 antigen binding sites. Which of the following statement best describes their targets? So we're looking here to the right statement, not the wrong one. Be careful, guys. Statement number A, 3 T cell antigens and 1 myeloma cell antigen. These are trispecific antibodies, guys. 1 T cell antigen and 2 myeloma cell antigens, or statement number C, 2 T cell antigens and 1 myeloma cell antigen. Please vote. And as I mentioned, we'll share the results with you at the end, of course, because this is a pre-assessment before the start of the educational activity, and we'll see the post-assessment. So with this, I'd like to close this introduction, and I would like to introduce Professor Marie-V. Mateos from the University of Salamanca, and she will address the very important topic of targeting Bcma. And as I mentioned in my introduction, today we have CAR T cells approved against Bcma. We have bispecific antibodies maybe to be approved soon, but also we have antibody-drug conjugates which target Bcma and which are approved. So Marie-V, thank you for joining us today, and the floor is yours.
Thank you very much, Mohamad, for the kind invitation, and it is actually a great pleasure for me to participate in this educational event focused on novel agents in multiple myeloma. And as you previously said, I am going to target on the novel agents targeting Bcma. And here you can see my conflict of interest. And also I would like to start with a question to the audience. What is your standard of care for the triple drug class refractory myeloma patients? Recycle previously used drugs if the patient was sensitive? Include the patient in a clinical trial? There is no worldwide standard of care available yet. All of the above are true. And please vote. And I see here the answer, and majority of you consider that all of the above are true. And this is completely correct, and this will give me the opportunity to move to the next slide in order to put in context that the triple class exposed, and especially the triple class refractory myeloma patients, are actually an unmet medical need. And here in this slide, you can see a couple of observational studies, retrospective for the mammoth, prospective for the locomotion, but both try to evaluate what were the rescue therapies for the myeloma patients already exposed to proteasome inhibitors, SNPs, and anti-CD38, and what was the outcome. And in these two studies, the main take-home messages are: point number one, there is not any standard of care for these patients. And in the locomotion, more than 92 different treatment regimes were used. And in terms of outcomes, the locomotion showed how the median progression-free survival was 4.6 months. And in terms of overall survival, the Mammoth study reported in the triple-class refractory population a median overall survival of 9 months. And this is the reason why we need novel agents targeting different antigens expressed on the surface of the plasma cells. And this is the rationale for Bcma that actually emerged as an optimal new target in multiple myeloma. You know that the Bcma is specifically expressed on the surface of the plasma cells. You have to know, and maybe you know, that the Bcma has a physiological function on the plasma cells, and Bcma promotes the proliferation of the plasma cells, promotes and supports the long-lived plasma cells, and it is involved in the production of normal immunoglobulins. And in addition, in myeloma, Bcma does contribute to create an immunosuppressive microenvironment. Bcma, in principle, with this mechanism of action, seem to be an excellent target, and this is what we are going to see throughout the next slide. And in addition, you have to know that the Bcma, is it possible to be targeted throughout the use of antibody-drug conjugates, CAR-Ts, or bispecific monoclonal antibodies. Starting by the bispecific monoclonal antibodies, Balentimab mafodotin was the first Bcma-targeted therapy approved for patients with multiple myeloma. And the rationale was 100 relapsed and refractory myeloma patients, all of them triple-class refractory, were exposed to Balentimab mafodotin single agent, an antibody-drug conjugated that targets Bcma, and it releases a cytotoxic mafodotin or monomethylauristatin F within the plasma cell, resulting into the killing of the plasma cells. But in addition, Balentimab mafodotin has also an immunomodulatory effect, and the antibody-dependent cell cytotoxicity, as well as the phagocytosis and the immunogenic cell death, are additional mechanisms of actions. As I previously told you, Balentimab was evaluated in 100 triple-class refractory myeloma patients. The overall response rate was 32%, what is maybe in line with the overall response rate expected for a new agent. But what is important to see is how majority of these response rate were VGPR rate or better, and in addition, the durability of the response was of approximately 1 year. And this translated also into a benefit in overall survival, with a median overall survival of 14 months. And these data were actually the rationale for the approval of Balentimab single agent. From the safety point of view, you know that we have to take care with thrombocytopenia, but especially with keratopathy, because of the cytotoxic released into the plasma cells. We have to incorporate ophthalmologists into our multidisciplinary team. Ophthalmologists have to evaluate the visual acuity as well as to explore the cornea with the slit lamp, but you have to know that majority of the patients will develop some degree of keratopathy, but majority of the patients will develop keratopathy grade 1 and 2. In principle, this keratopathy is not going to impact in the quality of life of the patients. This has to be the main objective, and it is quite manageable and reversible. The clinical development plan for Balentimab is very wide, and it is right now being evaluated in combination with different standard of care across the different lines of the treatment, since the first line of therapy to heavily pretreated myeloma patients. The second Bcma targeted therapy so far approved also by FDA as well as the European Medicines Agency has been ide-cel. And this is the Bcma CAR-T. You know that the structure of this drug of this therapy is different, and the rationale for the approval of ide-cel was more or less the same, because 128 relapsed and refractory myeloma patients after a median of 6 prior lines of therapy, and I would like to put as common denominator than 84% of the patients were triple class refractory, were treated with ide-cel, as you know, single agent, single infusion. And the efficacy was great because in the intent to treat patient population, the overall response rate was 73% and the complete response rate was 33%, indicating that these data are definitely better than those reported in the Mammoth or in the locomotion study. In terms of progression-free survival, overall it was of approximately 9 months, but it's true that if we focus on patients receiving the optimal dose, 450 millions, the median PFS was 1 year, and in terms of PFS by best response, if the patient achieved complete response, the median PFS was 20 months, and this is applicable to over 30% of the patients. And ide-cel resulted in a median overall survival of approximately 2 years, and again, this is clearly superior in comparison with the survival reported in the Mammoth and in the locomotion study. ide-cel is effective, and cilta-cel, I would say that is at least equally or more effective than ide-cel. It has been also recently approved by FDA and European Medicines Agency. The difference with ide-cel, we have to realize that the drugs are completely different, but you have to know that cilta-cel has 2 binding domains to Bcma on the surface of the plasma cells, and this can potentially increase the specificity. The rationale for the approval is again the same: 97 relapsed and refractory myeloma patients after a median of 6 prior lines of therapy, and again, 88% of the patients were triple class refractory, were treated with cilta-cel. And the overall response rate was 98%, indicating that basically all patients achieved some degree of response, but the stringent complete response rate was present in 83% of the patients. And in majority of these patients, the minimal residual disease was indeed negative. This has translated into a benefit in progression-free survival. At 2 years, the median PFS has not been reached, and 60% of the patients remain alive and progression-free. And from the overall survival point of view, 74% of the patients remain alive at 2 years. So it seems quite evident how both ide-cel and cilta-cel covered the unmet medical need we had in myeloma. But from the safety point of view, we have to consider the cytokine release syndrome, neurotoxicity, cytopenias, infections, and less frequent, but also B-cell aplasia and hypogammaglobulinemia, as well as a macrophage activation syndrome. And in addition, you have to consider that when a patient is eligible for CAR-T, the patient has to follow this roadmap, this journey, that it is not immediate, and you know that the CAR-T requires from manufacturing process, and this usually takes between 4 and 6 weeks. So the delivery process for CAR-T is more complex in comparison with Balentimab mafodotin. And in the middle, we have another possibility for targeting Bcma throughout the use of bispecific monoclonal antibodies. Bispecific monoclonal antibodies targeting Bcma and CD3 T cells. And these antibodies engage the T cells in order to be redirected into the tumor cell. There is here a long list of Bcma bispecific monoclonal antibodies. basically evaluated also in triple-class refractory myeloma patients, and it's true that these studies, majority of them are phase 1-2, in which patients are receiving different doses, but if we select the appropriate dose, you can see how the overall response rate ranges between 60 and up to 80 or 90%, indicating that these BCMA bispecific monoclonal antibodies are also very effective in this population. It's true that we don't have a solid information in terms of durability of the response or progression-free survival, but in principle, they will be also incorporated into our treatment landscape. One difference with BCMA CAR-Ts and in line with Balentimab mafodotin, bispecific monoclonal antibodies are off-the-shelf drugs, and definitely the eligibility criteria will allow to include patients under treatment with bispecific if they are older or less fit in comparison with CAR-T. And in the clinical trials, you can see how patients older than 80 years have been so far included in these clinical studies. And we have to consider the safety profile, cytokine release syndrome also present in majority of the patients, although less frequent than with CAR-T, neurotoxicity less frequent, but cytopenias and especially hypogammaglobulinemia is for me the most relevant BCMA bispecific related adverse events, and we have to take care about infections. How to proceed in the clinic? From my point of view, we are going to see patients eligible for whatever of these approaches is, because ADCs and bispecifics are off-the-shelf drugs, and definitely antibody drug conjugates today can be delivered in older patients, in patients even with some degree of renal impairment. And one of the main advantages for ADCs as well as bispecific antibodies is the possibility of being combined with other different immunotherapy strategies or with other backbones. And concerning CAR-Ts, as I previously told you, maybe is the most effective approach we have from all BCMA targeted therapy, but we have to consider the manufacturing process, the restriction in terms of patient eligibility as well as slot availability, and we have not observed any plateau phase yet in the progression-free survival curves. Today, these BCMA targeted therapy are going to be used in the triple-class exposed triple-class refractory myeloma patients, but you know that there are many phase 3 clinical trials going on, and my personal view is in the near future, this BCMA targeted therapy will move to first relapses, and of course, later on will take part of the first line of therapy. And I stop here. Thank you very much for your attention.
Thank you very much, Marie-V, really for a lovely talk. And as usual, you managed in such a short amount of time to give us a very comprehensive overview about the situation. And in this case, it was BCMA as a target and the different options and different tools that are being used to target BCMA, CAR T cells, antibody drug conjugates, but also bispecific antibodies. We will take already a few questions, if you allow me, Marc, to Marie-V. So we have a question from the audience about the expression of BCMA. Is the level of expression of BCMA on plasma cells of importance when it comes to the efficacy of these agents?
Well, in principle, the BCMA expression is necessary in order the drugs to work because this is the target. But in principle, the quantity of BCMA antigens expressed on the surface of the plasma cell is not so relevant. And in addition, you know that there are some strategies ongoing in order to increase the BCMA expression on the surface of the plasma cell throughout the use of gamma secretase inhibitor. But in principle, BCMA is quite specific from the plasma cells, And at the beginning, if the patient has not been previously exposed to another BCMA targeted therapy, I would say that it's quite constant, and all patients usually express BCMA on the surface of the plasma cells.
Thank you very much. Very clear answer. So actually, to echo this question, we have another question about when BCMA targeted treatments fail. Is it about the downregulation of BCMA or the mechanism of resistance is somewhere else?
Well, there are different possibilities. One potential mechanism of resistance is, of course, the loss of BCMA, and this has been described fortunately, in few patients after BCMA CAR-T. This is usually associated with the deletion of the chromosome 16, but it's not so frequent. But we have to consider this as a potential mechanism of resistance. Of course, it is possible just the BCMA to be shed, not disappear, but to be shed, and we can utilize a gamma secretase inhibitor in order to upregulate the expression. We have also to consider the potential generation of antibody anti-CAR-T as another potential mechanism of resistance, but we can't forget the T lymphocytes also as a source of resistance to the BCMA targeted therapy, the CAR-Ts, the T cell exhaustion, as well as the non-permissive microenvironment, overexpression of PD-1 and PD-L1 as a potential mechanism of resistance. So definitely, I think that we can't focus just on one mechanism of resistance, and the ideal situation would be to know what is the mechanism of resistance leading the lack of efficacy in each patient in order to try to generate strategies to overcome the resistance.
Okay, I'll take another question, which I think an interesting one from our friend, Dr. Sari Bygrave. Hi, Sari. What do you think is the ideal drug to be combined with Balentimab? We know today Balentimab, an antibody drug conjugate, is approved as a single agent, but if you have to combine it with other available agents, what do you think would be the best partner, Marie-V?
Well, I think that in principle, it would be ideal to combine with immunomodulatory drugs. And indeed, Susan Trudel from Canada presented the data from the Algonquin study in combination Balentimab with the pomalidomide with excellent results with a median PFS in a cohort of patients of 26 months. But I think that it is also possible to be combined with proteasome inhibitors, and the combination can potentially enhance the immunogenic cell death. And I think that this mechanism of action is also quite interesting. Maybe it can be also combined with anti-CD38 monoclonal antibodies. And we will see, because as I previously told you, there are, I don't know how many DREAM studies combining Balentimab with the different backbones, and since the beginning, to the late advanced stage of the disease. So I think that the good news is it is quite combinable with every PI, IMiD, or anti-CD38 monoclonal antibody.
Excellent. Thank you very much, Marie-V. And guys, don't worry. I know we didn't answer all the questions, but I'm keeping some of them until the end because some of them I think would fit very well in a nice global discussion with Dr. Raab. So now it is my pleasure to introduce Marc Raab from the University Hospital of Heidelberg in Germany. And Marc has kindly accepted to tackle the topic of CD38, but also SLAMF7. Remember, it used to be called CS1, by the way, a few years ago, as a target for therapy in multiple myeloma. Thank you, Marc, for joining us today.
Thank you, Mohamad, for the kind invitation, and Marie-V for setting up the stage and putting the bar so high after summarizing all the exciting BCMA data. So let's see. Yes, these are my disclosures. Yeah, so after all the fuss and a positive meaning about BCMA and many results from trials on targeting BCMA therapies, We should not forget about other antigens, and I think the most well-known in myeloma otherwise is CD38 and SLAMF7. So let me briefly update you on some more mature results now where we actually target SLAMF7 and CD38 with monoclonal antibodies. So there are recent data updated from the ELOQUENT-3 study with elotuzumab from Apollo and Bcma with different combination agents using daratumumab or isatuximab, both targeting, as we know, CD38. And then also give you a little bit of a outlook in the future, what happens on the CAR T cell space, so to say, with targeting SLAMF7 there, and also in the TCE, so T cell engager field, now also involving CD38 as an antibody, as an antigen, including in this case with a trispecific antibody. And of course, we do have, I think, in myeloma right now, two challenging patient populations, so to say, cohorts. These are the ones with LEN exposure, especially with LEN refractory disease after the first or second line. And then, as Marie already showed, the triple class refractory, even penta drug refractory patients where we need novel agents and novel targets. So let me start with the first group, so the LEN exposed patient group, and just an update on the ELOQUENT-3 study. You all know the design. It has been published quite a few years ago. PomDex as a background, as we all know it, and added elotuzumab in one arm compared to the classical PomDex control arm. Patients had to be at least pre-treated and relapsed from lenalidomide and at least 2 prior lines of therapies. And the PFS here was clearly superior with elotuzumab combined with pomalidomide, dexamethasone compared to PomDex alone. As you can see, 18 months, 34% compared to 11% of progression-free survival of the patients. Quite recently now, there's new data in terms of overall survival available, and you can see, and this is of course very important in the context of all the new agents, that those patients here who had received PomDex alone had a 36 months overall survival of less than 30%, whereas you see a continuous superiority of the triple combination with elotuzumab and resulting in a hazard ratio of 0.6. So clear benefit for the overall survival here of elo-PomDex compared to PomDex. Then let's switch to the carfilzomib combination with isatuximab, so a CD38 PI combination in the Bcma trial. You also know very likely the trial design, 3:2 randomization. Carfilzomib, as we all know it from the Endeavor trial, with twice weekly 56 milligram per square meter of carfilzomib, and then in the experimental arm added isatuximab bi-weekly at the end after one cycle of weekly treatment. 1 to 3 prior lines of therapy. Quite a few patients were LEN exposed and refractory. And you see here the most recent update on the PFS of ISA-KD versus KD, and you will see impressive medium progression-free survival of more than 35 months if you add a CD38 antibody to carfilzomib, in this case isatuximab, compared to the KD arm alone, which already went quite well for the patients compared to the Endeavor trial with 19 months median progression-free survival. So patient hazard ratio of 0.58, clearly showing benefit for CD38 plus carfilzomib in this patient population. And it also worked in those patients who had previously been exposed to lenalidomide and who had been refractory to lenalidomide, as you can see here. Then, last but not least, on these update sections, so to say, switching to daratumumab as probably the most well-known and most frequently used CD38 antibody due to very early or way earlier approval. And now a combination with PomDex, you know, the Apollo trial, adding daratumumab to PomDex compared to the classical PomDex regimen. Also patients with exposure exposure to LEN and a proteasome inhibitor before. Most of these patients were actually LEN refractory, or many of them. And you see also a clear benefit, again, hazard ratio 0.6 with a PFS here of about 12.4 months if you add daratumumab, in this case, to pomalidomide. So I showed you that all these antigens are absolutely important targets, and we can use monoclonal antibodies against those targets, adding to our standard of care drugs, as we do now in a daily routine, and this results in clear patient benefit. Also given the relatively moderate added toxicity and side effects by these monoclonal antibodies. So let's switch to the very challenging patient population, the triple class refractory. And as Marie has pointed out, CAR T cells are certainly a major step forward with unprecedented response rates and PFS rates for the time being. And there are, of course, many other CAR T cell therapies or CAR T cell constructs under development for all kinds of targets, as shown here on the right side. And I have to acknowledge the slides from Michael Hudacek, who sent me them kindly and provided them. And I will, of course, now, being the title, SLAMF7 as the target also for CAR T cell therapy in emphasis. And I can show you the overview of actually what Michael Hudacek has developed from at our colleagues' site in Würzburg. Meaning SLAMF7 CAR T cell therapy. And it is different, not only in terms of target, but also in terms of manufacturing process, and specifically the setup, how they built this CAR T cell construct. They used a virus-free manufacturing process by Sleeping Beauty transposons to get this into the T cell, which they have now regulatory approval for this kind of production process, and we're able to now translate this into a clinical trial in an EU consortium with many countries in the EU participating. Before that, of course, they have shown that in vitro as well as in mice, the SLAMF7 CAR T cell therapy works quite specifically and nicely, as you can see here. So the mice overall showed a absolutely reduction of the, you can see here this as well, of the tumor load compared to the control mice or the untreated mice from both approaches. So it is specific and is effective in the preclinical setting. And as I said, this is now being explored in a phase 1 trial. And if I'm not mistaken, I think they have reached now the 3rd dose level without having had any DLTs so far. Otherwise, it is the typical lymphodepletion setup, and then, of course, the follow-up of the patients, and they aim for a quite short vein-to-vein time in about 16 days, meaning that one obstacle could be overcome potentially by such a manufacturing approach that Marie has alluded to. So the quite several weeks we have to wait between apheresis and the actual CAR T cell therapy for the patient, especially in the refractory setting. And last but not least, I want to show a few slides on development that is currently ongoing with a tri-specific antibody that targets CD38 as a target in myeloma. Don't have to put any further words on this target in myeloma, of course. Combining CD38 with CD3 on a T cell in a T cell engager construct, and adding a third antigen that is targeted by this tri-specific antibody, making it a tri-specific antibody, namely a CD28. CD28, we all know, actually also from a CAR T cell therapy and constructs, is a co-stimulatory receptor that regulates T cell activation, and actually, especially in late stage myeloma, there's a substantial fraction of myeloma cells in myeloma patients who actually do express CD28 as well on the myeloma cell, making the initial question that was answered by you actually a little bit tricky to answer. So as you can see here, it's a clear benefit in the mice model for these trispecific antibodies, and more importantly, and because specifically, if you can compare here in 3 different myeloma cell lines, all of them have to say also co-expressing CD28 together with CD38 on their cell surface. And what you can see here in blue is the added benefit of a trispecific antibody, CD38 binding and CD28 binding, of course, in addition to CD3, compared to the same antibody, but with a CD28 knockout construct, so only binding to CD38 and CD3. And you clearly see that that there was about a 30 to 100-fold increased efficacy of the trispecific antibody compared to the same construct, but only targeting 2 antigens at the same time, CD38 and CD3. This is now also being explored in the clinical trial, a phase 1 clinical trial. To the best of my knowledge, the MTD has not been reached yet, and of course will then be explored in combination therapies later on. With this, I want to summarize. So we have 4 patients in second, third line, a huge portfolio of different combinations, all of them, not all, but many of them targeting either CD38 or SLAMF7 in combination therapies. The challenge will, of course, be that many of these agents and combinations are moving and have been moved to first-line therapy. So in the future, we will face refractory or at least relapse refractory patients after these kind of drug exposures already. So we need the novel therapies then in the future already in second and third line. But for the time being, I think we're well equipped for these lines of therapies. And for the triple class refractory, as Marie has shown with CAR T-cell on Bcma, we have more underway in development targeting CD38, SLAMF7. Same holds true for the bispecific antibodies. And you know there's more like GPRC5D, FCRH5 as targets, which also have been have shown very effective response rate and duration of responses, and now maybe potentiating even this approach of the TCEs now with trispecific antibodies. With this, I am happy to take questions and probably lead over to the discussion by Mohamad. Thank you very much.
Thank you very much, Marc, for really a very beautiful talk. And again, you nailed it very nicely, and you highlighted the very exciting developments we have today in the field of multiple myeloma. And this is really amazing because I'm not aware of any bispecific antibody being approved so far, and we're already talking about tri-specific, antibodies. This is really fascinating, guys. Okay, so we will take a few questions and we'll launch the discussion. And actually, I kept one question which I was supposed to ask in the beginning, and this is from a member of the audience asking, why did you choose specifically CD38, SLAMF7, and Bcma to be addressed in this webinar? And for instance, let me, I would say, expand on this. Let me start with you, Marc. Why CD38 and not CD138, let's say? What are the reasons behind, what is the added value of CD38, for instance, compared to CD138, which is also, Sendican-1 is expressed on plasma cell?
Well, that's a tough question. I mean, CD38 is absolutely a proven, as I have shown, right, a proven antigen to target in myeloma. It is very robustly expressed in myeloma cells, and I mean, little on other cells, as we all know, and obviously it does work. It is proven. There have been approaches targeting CD138, for example, with ADCs and other approaches. So far, the clinical data, interestingly enough, have not been convincing. So I think for the time being, absolutely CD38 is the proven target in terms of approval also, agents that have been approved for therapy, and now, of course, being added, I would not say challenged, but being accompanied by BCMA for all these active novel immunotherapies. And the same holds true for SLAMF7. I mean, we know that SLAMF7 is a robust target on myeloma cells. We know that it's almost expressed on in every patient. And we have experience of several years now with targeting SLAMF7. So, I mean, this is absolutely, I think, the most established antigens, notwithstanding, of course, that targeting, as I already mentioned, GPRC5D or so, is of minor importance, right? But I think for those antigens, we do have the biggest bulk of data and the most experience. That would be my answer. It would be interesting to see and hear what Marivi says.
So my question to Marivi will be actually, let's focus on BCMA. Why BCMA is interesting, and I guess because we had also a comment about this, is that actually the knockout mice can live well without BCMA. So I guess this is one of the arguments. Maybe you can expand on this, Marivi.
Well, definitely BCMA. Well, when we try to find new drugs effective for patients with multiple myeloma, we have to try to find or to look for specific targets, and as much as possible only express on the surface of the tumor cells. And this is one of the main differences with CD138, to which Marc previously alluded. It is expressed on other cells, and indeed the first clinical study reported quite a high toxicity. So this is one of the reasons why BCMA emerged as a new target. It is quite specific from the plasma cells, and it is involved in the proliferation of the plasma cells. And it is also involved in the pathophysiology of the myeloma, because it does contribute to create the immunosuppressive environment. So this is the theory. And when they decided to generate strategies like conjugated bispecific or CAR-T, well, the efficacy is there. It's the same situation for CD38, to which Marc alluded. The efficacy is there, and I think that it emerged, and BCMA has been consolidated as an optimal target. Is it possible to live without BCMA? It is, and indeed we have patients treated with BCMA targeted therapy for many years, and the most relevant effect we see is the severe hypogammaglobulinemia. So we can live without BCMA. I'm not so sure if we can live forever without immunoglobulins. We can, but we have to take into consideration infections, especially opportunistic infections, viral infections. But I think that this is the second page of the history. I think that now this BCMA targeted therapy are being evaluated in heavily pretreated myeloma patients until progressive disease. The efficacy data are great, and the next step is to optimize, maybe throughout its use as a fixed duration of therapy, in order to not impact continuously on the expression of the BCMA on the surface of the plasma cells.
Excellent. Thank you very much. One question for both of you guys. This is from Dr. Vedata Slan. Can we combine actually the anti-CD38 monoclonal antibodies like cetuximab or daratumumab with bispecific antibodies to have a sort of a combination? Because again, I think you mentioned in your talks that bispecific antibodies for the time being are being used as single agents in the most relapsed refractory advanced patient. But as far as I know, I think we can combine them. Maybe you can both expand on this issue of combining actually monoclonal antibodies plus bispecific antibodies.
Yeah, so I think that this is an excellent opportunity to combine. As you mentioned, we have already some clinical data showing how the combination resulted in a higher overall response rate in comparison with CD38 or BCMA bispecific monoclonal as single agents. And in addition, from the pharmacodynamic point of view, it is quite interesting to see how after the DAR administration, it is going to be a depletion in the CD38 positive T cells, but the use of teclistamab later on at the beginning of the cycle 2 is able to increase again the population of the T cells that are definitely necessary in order to be engaged, in order to be redirected into the tumor niche. So there are evidence from the clinical point of view, from the pharmacodynamic point of view, and this is applicable to teclistamab, but this is also applicable to talquetamab. And I think that this is one of the main advantages for bispecifics as well as for antibody drug conjugates.
Excellent.
Yeah,
Any
I
comments on
absolutely
this, Marc?
Yeah, no, I mean, as Marie said, there are clear rationales why combinations should make sense. We will see whether it is really resulting in superior outcome in a clinical setting. This is too early to say. And of course, since especially bispecifics will be combined, I would say with every standard of care drug and combination we have, I mean, we will get more and more data in the next years to come, whether all these preclinical and immunological rationales actually will pan out in a superior patient outcome and survival.
So one question, and again, I don't want, it's a difficult one, I think. And this is from Dr. Gara Manian. I hope I'm pronouncing well. And this is about among the different options that both of you described and mentioned, what would be your favorite option or combination for patient with extramedullary disease? Mary V.
Well, I am smiling because you know that this population is always underrepresented in the clinical studies. And indeed, you had the opportunity to see how in all these clinical studies, the sample size are 100, 128, 30. So at the end of the day, the information we have is quite limited. But it is interesting to see how whatever of these novel approaches seems to work at least partially in patients with extramedullary disease. And this is applicable to Bcma CAR-Ts. This is applicable to some clinical cases reported treated with Balentimab mafodotin, and the same for bispecific monoclonal antibodies. We have to assume that these patients treated with these novel modalities have previously received 7 or 8 prior lines of therapy. So definitely if we see some activity, and I think that this is a good signal, although we have to recognize that extramedullary disease does continue being one of the most challenging high-risk features in multiple myeloma, and we will see with more data. Fortunately, I have to say that some of these clinical trials conducted with bispecific monoclonal antibodies allow specifically to include patients with measurable disease by extramedullary disease. That this is extremely important because as soon as we move to late advanced stage of the disease, the myeloma is less and less secretor, and sometimes we see more and more extramedullary disease, and patients can't be included in clinical trials. But I think that we are going to have more data. I would like to be optimistic, although also realistic, and extramedullary disease is a challenging situation.
Thank you, Mary V. But again, I think we can be optimistic. I feel that we need to be optimistic, and we will win this fight at the end. One question for you, Marc, is about targeting 2 antigens with CAR T cells. And I think it's a relevant question because you mentioned SLAMF7 as being tested as a target in CAR T cells, but also we have, of course, the BCMA as a target with CAR T cells. What about the dual targeting? And I guess by dual targeting, you can either manufacture, design one CAR T cells targeting the 2 antigens, or actually have a sort of a sequential infusion of 2 CAR T cells. I know this is maybe like science fiction, but can you comment on this?
At least it's not fiction, because I mean there's very limited, but I think there's a few data on that. I think, for example, Adam Cohen and UPenn and so on, they have done similar things, and there are, as far as I know, there are dual targeting CAR Ts, so actually really targeting two antigens at the same time with the same CAR T cell under development. I think so far we cannot say convincingly, because the clinical data are pending, that this approach are superior to what we have now and will be superior to the combination of these single targeting agents, so to say, in combination with standard of care drugs, but is an absolute fascinating field with the idea to at least delay the antigen escape, so to say, if this is the mechanism of resistance. However, as was discussed before after Marivi's talk, I mean, we have to learn way more about the immune system and the microenvironment, how that contributes to the resistance mechanisms, and whether their dual targeting approach is a beneficiary or not. But at least there will be data, and I think it's a very exciting field to see the outcomes on that. Yeah.
Excellent. Thank you very much. We'll take one question before the concluding remarks. Mary V, who are the patient today to whom you're giving Balentimab as a salvage therapy?
Yeah, for me, basically the relapsed and refractory myeloma patient already exposed to PI, IMiD, and anti-CD38 monoclonal antibody. Maybe because Belamaf is an off-the-shelf drug, we can consider aggressive disease, but on the other side, because of their route of administration, IV in only 30 minutes, and the safety profile, thrombocytopenia and keratopathy, maybe a more elderly population will be eligible for receiving Belantamab mafodotin. And also in my clinic, I usually consider the renal function, because elderly patients usually present with some degree of renal impairment between 30 and 40. Is quite common to have patients in this situation. These patients are not eligible in principle for bispecifics or CAR T, and I think Belantamab mafodotin is an excellent opportunity. So I will move today, if the three options would be available, for less fit population, more elderly, with a good performance status, or patients with some degree of renal impairment.
Excellent. Thank you very much. This was really a lovely discussion. Here, guys, I would like to come back to our assessment questions for this educational activity. And this is, again, question number 1. And actually, most of you got it right here. So what is the wrong statement actually when about CAR T cell? They are less effective than bispecific antibodies or actually antibody drug conjugate for the treatment of multiple myeloma. Treatment only requires a single infusion, and not all patients with refractory multiple myeloma are eligible for treatment. So, well, since most of you got it right, actually there was little change, I would say, in the percentage of answers. More than 80% got it right. Of course, no one can claim today that CAR T cell treatments are less effective than bispecific antibodies or antibody drug conjugates. Of course, we use them as single infusion. And of course, not all patients are eligible to these treatments. Question number 2, which is a little bit more difficult, guys. CAR T therapies for multiple myeloma are currently under development for which of the following target antigens? So we heard from Mary V all the CAR T cells against Bcma, and they are approved actually, by the way, whether ide-cel or cilta-cel. But here, what is being developed? CAR T cells against PD-L1, SLAM-F7, or CD40. It was a little bit more difficult than the previous one, guys. Okay, so fantastic, because when I asked the question before the educational activity, actually we had 25% of the colleagues who got it wrong, but Dr. Marc Raab was very clear. And actually today, now we have 100% getting the right answer. This one was much more difficult, guys, and the majority got it wrong. So I hope now you will get it right. And this is about tri-specific antibodies. So they have 3 antigen binding sites. And which of the following statement best describe their targets? And again, it was clearly mentioned in the talks. Is it about 3 T-cell antigen and 1 myeloma antigen? But that gives you 4, guys. 1 T-cell antigen and 2 myeloma cell antigen, 3. 2 T-cell antigen and 1 myeloma cell antigen. Please vote. Okay, fantastic. Well, it has improved significantly because the right answer actually is that today, under development, we have tri-specific antibodies targeting 2 T-cell antigens and 1 myeloma cell antigen. So before the start of the educational activity, we had 30% who got it right. Now we have more than 60%. So I think we still have some work to do when it comes to these tri-specific antibodies. They are very complicated, but this is really fascinating. Well, guys, thank you. Well, thanks to you, Marie-V. Thanks to Marc. Thanks to the superb audience we had today. And thank you for all your questions, for all your comments. I do apologize that we were not able to take all the questions and comments, but for the sake of time, we have to make choices. So thank you very much for being loyal to this really nice special webinar and in general to the ICH activities. Please remember to complete the evaluation form. Your feedback is really, really important to us. So with this, I'd like again, thank you all for your attention. I hope you have enjoyed it. I have personally learned a lot from Dr. Raab, from Dr. Mateos, and from your questions and your comments. And as usual, I wish you a good evening, a good night, a good day, a good afternoon, wherever you are. Please stay safe and keep well. Thank you very much and see you soon.